So good afternoon, everyone. As I'm always privileged to have you on this channel, and I'm always at your service. And today, since it is Easter of 2024, I also wish my viewers the happy Easter. So we are going to continue talking about lipids. And if you have not watched the first video of introduction to lipids, first watch it before you watch this. It will help you so much. So let us get started on the digestion, absorption, and metabolism. We want to see if you eat lipids, how are they digested in the GIT? And how do they cross the GIT into the blood saturation? And how does the body utilize it to provide energy? Thank you so much. Let us get started. So digestion, we want to begin with digestion of lipids, whereby digestion of lipids, of course, as usual, it begins in the mouth, whereby in the mouth we have little digestion of lipids, whereby we have both physical and chemical. And physical digestion of lipids in the mouth is by action of teeth, which breaks down bigger food into smaller droplets. Then chemical, we know that there is production of saliva, and this saliva contains an enzyme called lipase. This enzyme, let me use a different color. This saliva is called lipase. And this lipase is the one that catalyzes the hydrolysis of triglycerides, or what we call triacylglycerol. It catalyzes the hydrolysis of triacylglycerol, which we call tags or triglycerides, to free fatty acids, to 2 free fatty acids plus MAG. MAG is known as monoacylglycerol. What are we meaning here? And this digestion in the mouth is minimal. It's very minimal. And this lipase acts on only short chain fatty acids. It works on short chain tags. And it just accounts for like 10%, which is minor. Then from the mouth, this food is swallowed into the stomach. And in the stomach, we have production of gastric juice. And this gastric juice contains hydrochloric acid and gastric lipase. Whereby hydrochloric acid is the one that kills any bacteria that enters with food, any pathogen, and also provides acidic pH. Then the production of gastric lipase in the gastric juice, it also works on the short chain tags, short chain triacylglycerol to monoacylglycerol plus 2 free fatty acids. But as I told you that the chemical digestion in the mouth and in the stomach is minimal. It does not go beyond 10% and it only acts on the short chain triglycerides. So the long chain triglycerides cannot be digested in the mouth and the stomach. That's where from the stomach we move into the pancreas, into the duodenum, and in the duodenum we have there the pancreas, whereby the pancreas within the duodenum, it releases pancreatic juice, and this pancreatic juice Maybe the first thing before production of pancreatic juice, how is it triggered? We normally say the presence of food chyme, presence of acidic food chyme in the duodenum, it triggers the production, of cholecystokinin hormone. This is the hormone, and this cholecystokinin hormone, it does two roles. So the presence of food chyme, acidic food chyme, it is detected in the duodenum, and the duodenum, we have there what we call CCK cells, which releases cholecystokinin hormone, which does two roles in digestion. Whereby cholecystokinin goes to the gallbladder, can go to the gallbladder, and in the gallbladder, it stimulates the gallbladder to secrete bile. Then another function of CCK hormone, this hormone is abbreviated as CCK. Another function or role of cholecystokinin is it goes to the acinar cells of the pancreas, and stimulates them to release pancreatic juice. As we normally see that if this is my structure of the liver, the structure of the liver. In the liver, we have this falciform ligament, then we have the left hepatic and the right hepatic meeting at the gallbladder. This is the gallbladder, then it joins into the bile duct, and the bile duct joins the duodenum at the junction known as ampulla of Vater, and at this ampulla of Vater, we also have our pancreas connecting there. So what happens, the presence of food chyme, I have talked about acidic food chyme, triggers these CCK cells. The CCK cells secrete cholecystokinin, and one of the cholecystokinin comes to the gallbladder in the liver and stimulates production of bile and bile salts. This bile contains bile salts. Then we see another one goes to the pancreas, another CCK goes to the cells in the pancreas, and it triggers production of pancreatic juice. And all these ones work hand in hand for the digestion of lipids, because we know major digestion of lipids occurs in the duodenum. So what happens is that after production of bile, and this bile containing bile salts and bile acids, these bile salts and acids will go and cause what we call emulsification of fats. They solubilize the fat. Because when we eat the triglyceride, it is a big globule. It cannot be hydrolyzed by the enzyme. So what bile does is to come and break down the big fat globule, the big fat, into smaller droplets, to smaller lipid droplets, which we call micelles. And this process is known as emulsification. Whereby it is action of bile salts. Presence of bile salts, it emulsifies the fats. How? By breaking down a big fat droplet into smaller lipid droplets called micelles, which can easily be digested by the pancreatic lipase. Because we know that these micelles, when we form smaller droplets, they increase the surface area for the action of the enzyme. So after breaking down into smaller droplets, now the pancreatic juice which was triggered contain pancreatic lipase. Contain another enzyme known as the phospholipase. Phospholipase, let me write it here. Phospholipase A2, and it contains another enzyme known as cholesterol esterase. So there are major two enzymes in the pancreatic juice, three. The first enzyme is the pancreatic lipase, Then we have phospholipase A2 and cholesterol esterase. As I told you in the beginning, the fats we eat, the food we eat, contains triglycerides, contain phospholipids, and contain cholesterol esters. So we need to hydrolyze them. So we shall see these enzymes after breaking the big fat globule into smaller droplets called micelles to increase the surface area for action of these enzymes. So we are going to see these enzymes catalyzing these fats, whereby in presence of pancreatic lipase, we are going to see conversion of triglycerides, which we call tags, to two free fatty acids plus monoacyl glycine. And this is the hydrolysis reaction, so it occurs in the presence of water. Then we have another enzyme known as phospholipase A2. Phospholipase A2. We have another enzyme, cholesterol esterase. These are the enzymes in the pancreatic juice. Phospholipase A2, it catalyzes the hydrolysis of phospholipids in the food. In the food we eat, we have phospholipids, and they are catalyzed or they are broken down to lysophospholipids. Lysophospholipids. Then the cholesterol ester in the food, cholesterol esters in the food are hydrolyzed by cholesterol esterase enzyme in the presence of water to form free cholesterol. And as I told you that, as we know that lipase and the gastric lipase are acting on the short chain fatty acids, for this enzyme, pancreatic lipase acts on the long chain fatty acids. Then this one acts on the phospholipids and this one acts on the cholesterol esters in the pancreas. So this is where the major digestion of lipids occur in the duodenum. Occurs in the duodenum. So from the duodenum, we know another part of which is the part of the small intestine. Now we move to the ileum. We move to the ileum, whereby in the ileum we see only absorption. So digestion of fats stop here. It stops here. Whenever we go to the ileum, now we talk about absorption. So in the ileum, we see absorption of fats. So digestion, there is no digestion in the ileum. No digestion of fats in the ileum. Digestion of fats stops in the duodenum. In the ileum, we see absorption. And how are these fats absorbed? How are they absorbed into the blood? If I can draw a rumen here, this is the intestinal rumen. This is the rumen of the ileum, and this is the intestinal mucosa. Intestinal mucosa, that is the intestinal wall. You can call it intestinal wall. Intestinal wall. So what happens if I have formed products? I have free fatty acids. Hope you're seeing them. I have monoacyl glycine. I have lysophospholipid, and I have free cholesterol. These are the products of fat digestion. So these are products, the free fatty acids, the monoacyl glycine, the lysophospholipid, and the free cholesterol all combine together. And when they combine, so here we are looking at, in the ileum, we are looking at the absorption, as I had told you. And we have found that these products of digestion, the free fatty acids, the monoacylglycerol, the lysophospholipid, and the free cholesterol, they combine with fat-soluble vitamin. The fat-soluble vitamin A, D, E, and K combine with the products of digestion. And after combining, they form what we call mixed micelle. Mixed micelle. So this is the product we have formed, which has reached in the ileum. That in the ileum, we get these products of digestion from the pancreas, joining together with the fat-soluble vitamins, and to form what we call a mixed micelle. Why are the fat-soluble vitamins combining? Is that they are the one that are going to enable this simple diffusion of fats into the intestinal wall. So I told you I have zoomed the intestinal wall. So this intestinal wall is starting from here up to here. This is the intestinal wall. So this intestinal wall, after the fat-soluble vitamin is combining to form a mixed micelle, they cross by simple diffusion. This process is diffusion. They cross the intestinal wall by diffusion, and they enter into the walls of the ileum, isn't it? So we shall find our products, the free fatty acids, the monoacylglycerol, the lysophospholipid, the free cholesterol, together with their fat-soluble vitamins. Together with the fat-soluble vitamins, which combined the vitamin A, D, E, and K, they combine together into the intestinal wall, and the essence of digestion here is to enable them to cross. Can you imagine? So the whole digestion process we are looking at is to break down into smaller portions so that they can enable crossing of these fats by simple diffusion. After crossing and reaching into the intestinal wall, they are converted back. We shall see the free fatty acid is combining again with the monoacylglycerol to form triglycerides. We are going to see lysophospholipid being reconverted to phospholipid. We are also going to see free cholesterol being reconverted to cholesterol ester. So the essence of digestion is to enable crossing from the intestinal lumen into the intestinal wall. When they reach in the intestinal wall or mucosa, they are the major word is that they are converted. And we can see here how they are converted. If I can enlarge this lumen, let me enlarge it further. So we are going to see free fatty acids and monoacylglycerol being reconverted back to triglycerides. We are going to see lysophospholipid being reconverted to phospholipid. Then the free cholesterol is reconverted to cholesterol esters. So here the term is reconversion. When they cross by simple diffusion, they are reconverted. Reconverted. And this reconversion does not need the enzyme. It doesn't need enzymes. So what we were breaking down here was to enable crossing. Like the way Moses created a dry path on the Red Sea for the Israelites. So it was creating the way for the crossing of these triglycerides, isn't it? So after forming back our products which we ate in the food, these products which we have reconverted combine again. They combine together with a protein. And this protein which combines there is called chylomicron. This chylomicron is a lipoprotein. And you know the function of lipoprotein is to transport. They are like buses and taxis. They transport lipids. So this, after reconversion, we are seeing triglycerides, the phospholipids, and the cholesterol whereby triglycerides account for 90%. 90% of this is the triglycerides, whereby phospholipids are covering 5%, like 1 to 2%, and lipids are accounting for 5%. The cholesterol is accounting for 5%. So the chylomicron joins them to enable them to move. So after putting a chylomicron on these products, they are transported via the lymphatic system. Remember the microvilli of the intestinal lumen of the ileum, they have the lactive. And this lactive is the one that connects it to the lymphatic system, isn't it? So these fats, the ones which are with long chain, they go into the lymphatic system of vessels, and then they are transported to the vessels near the heart. Those are the thoracic ducts. Thoracic ducts, which are vessels near the heart. So after reaching into the thoracic ducts, these thoracic ducts are the ones that supply these lipids together with the chylomicron, which is transporting them into blood saturation. They enter into blood saturation. And from blood saturation, when they reach into blood saturation, then they are transported by blood to the majority of them go to adipose. In case there is no need of energy, when you are not starving, they are taken to adipose and store away. Then another percent, in case we need energy when we are doing strenuous exercise, and we need more energy in skeletal muscles, these fats are taken to skeletal muscles, where they are used to provide energy. Then the remaining part, which we call the chylomicron remnants, they are taken to the liver. The chylomicron remnants are taken to the liver. So we see these remnants being taken to the liver. So this is what we call absorption of fats from the intestines into blood saturation, then from blood saturation to the adipose and skeletal muscles. Where we have seen that the products of digestion combine with fat-soluble vitamin to form mixed cells, and which enable them to cross the intestinal walls into the intestinal mucosa. And within intestinal mucosa, we see them being reconverted to their original form. After being reconverted, they combine with the chylomicron, which is a lipoprotein, which enables them to be transported to the lymphatic vessels, then from the lymphatic vessels to the thoracic ducts, which are the vessels near the heart. Then from the vessels near the heart, they supply them to the general saturation, where they are taken to the adipose, majorly skeletal muscles. Then the remnants, the little portion, is the one which is taken to the liver. This is the end of absorption. Thank you so much for listening. In the next video, we are going to talk about metabolism. How are these fats utilized in the body? Thank you so much for listening. Always remember to like the video. Thanks so much.